Hi Year 11s and 12s, welcome to Stage 2 Biology. This is going to be the very first PowerPoint in the very first unit, which we're talking about DNA and proteins. This PowerPoint is going to be split into 3 different videos, and we're going to be starting with DNA structure. This will go along with the workbook that you have for school, which is your SASTA workbook, and this will be Unit 1.1. We'll start the video as always in the same way, where you've got a need to know. So these are your SACE requirements and scientific understandings. I won't go through these on the slide and on the video, but if you'd like to write these down, which I'd recommend, pause the video here, write them down and tick them off as we go through them in the video. But let's actually start some of the content now.
I see a lot of new faces, but you know the old saying, out with the old, in with the nucleus. Now we'll begin by... Oops.
So, DNA and the cell. What we've understand from biology in the previous years you've studied, year 8, 9, 10, 11, is that the cell is the basic unit of life. All living things are made up of cells. And inside those cells, all of the life processes are carried out by macromolecules, and those macromolecules are called proteins. Now, a macromolecule, if you break down the word, macro means large, so large molecules. In biology, there's a lot of that. You need to understand the meaning of words. So, proteins are large molecules that carry out the life processes inside of cells. Now, proteins have many different forms. And the structure and the function of a particular protein is determined by the genetic information in a cell, which is all stored and transmitted by nucleic acids. Now, the nucleic acids that we're talking about are DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. DNA stores and transmits the genetic information the same way in every living organism. It's also described as a helical double-stranded molecule. Okay, so it goes around in a helix and it has 2 strands. I won't play this video to you on this video, but this is crash course number 10 on DNA structure. We will watch this in class. This will also be on Doing Science Right under the useful video sections of the Year 12 SACE page if you'd like to watch it beforehand. Nucleic acids. So nucleic acids are polymers. So a polymer, for those that don't study chemistry, is a large molecule made up of many small molecules that are exactly the same. So they're a small molecule that repeats itself over and over again, and those small molecules are called monomers. So nucleic acids, which are polymers, are made up of nucleotides, which is the monomer in this case. Now nucleotides, as we can see here, okay, or also here, consist of 3 different chemical groups. They're made up of a sugar, which in DNA and RNA is pentose sugar. So we can see here pentose sugar. They're made up of a phosphate group. Over here we've got the phosphate group. And they're also made up of a base, one of 4 different bases. There's actually 5 bases, which we'll talk about in the next slide. But DNA has only 4 bases and RNA only has 4 bases, but they each have 1 different to each other. The phosphate group and the base are both chemically bonded to the sugar. So the sugar's in the middle, the phosphate group's generally drawn on the left, and the base is generally drawn on the right. Now, it's a bit hard to see under this video, but we will see it in class as well. You should always, especially at the stage 2 level now, and in an exam it'd be required, you draw your phosphate group as a circle, your sugar, because it's pentose, as a pentagon, and your base would be drawn as a very irregular shape up here, which I'm trying to follow with my mouse. Hopefully you can see that on the screen. So, what are the DNA bases? I've said there's 5 different bases. What are they? So, the first 3 we'll look at are the bases that are found in both DNA and RNA. So, adenine, represented by an A, guanine, represented by a G, and cytosine, represented by a C, are all present in both DNA and RNA. DNA also contains thymine, which is represented by a T, And RNA also has uracil, which is presented as a U. Okay, now that's one of the main differences between DNA and RNA. RNA does not contain thymine. Every time there'd be a thymine in DNA, that would be shown as a uracil in RNA. Okay, now those bases are really important to the structure of DNA, which we'll look at in the next slide. But before we move on to that, the actual structure of DNA, it's a molecule that's made up of 2 polynucleotide strands. Okay, so we've got our 2 strands here wrapped around each other, or you can see in this diagram here, they've been unwound. Okay, and we can actually see one strand here, another strand here. Okay, the sugar and the phosphate groups of DNA are chemically bonded to each other, and then that repeats over and over again. So we have phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. On the other side, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay, because that's constantly repeating in that strand over here, and then you have the bases on the inside, we call this the sugar phosphate backbone, because it's the backbone of DNA, and it's made up of sugar and phosphate. The way that DNA arranges itself is that the nucleotides will always arrange themselves so that the base face inwards. So the sugar and phosphate groups on the outside, and the base is on the inside. The 2 strands will always run anti-parallel to each other, so you don't need to understand why they're called 5' and 3', but we call different ends 5' to 3', and DNA is always created in the 5' to 3' direction. So this strand's got phosphate, sugar, phosphate, sugar, phosphate, sugar, going from 5 to 3, whereas this side goes in the opposite direction, anti-parallel, 5' to 3', phosphate, sugar, phosphate, sugar, phosphate, sugar. The reason for that is so that we can actually get these bases on the same side, and they can actually bond together. Okay, we'll talk about why they have to be like that, and these 3 dots and 2 dots, 3 dots will make sense in a second. Okay, the way the DNA is structured is every 10 bases, the DNA will wind up on itself. That's why we refer to it as a helix. Okay, and we refer to it as a double helix because there's 2 strands. Those 2 strands are held together by these 2 bases at each time. So all the bases hold the 2 strands together, and they're held together by weak forces called hydrogen bonds. Okay, those hydrogen bonds keep the complementary bases together. Now the word complementary there is important because each base will only go with one other base. Okay, the bases will always pair up in the exact same way, and that gives order to the structure of DNA. So how do they pair up? Well, adenine will always pair with thymine. Now A with T, so straight lines with straight lines. Cytosine will always pair with guanine. Okay, curvy with curvy, C with G. They're both made out of curves. Both letters are made out of curves. So straight with straight, curvy with curvy. That's how you remember which one goes with which. Adenine with thymine, cytosine with guanine. Why do they pair up that way? This won't be in tests and exams, but it's useful information to provide in tests and exams to help show your understanding, is that the reason that adenine and thymine go together is because they both have space for 2 hydrogen bonds. Cytosine and guanine go together because they have space for 3 hydrogen bonds. You can't form 3 hydrogen bonds with thymine and adenine, so they can't connect together. So guanine and cytosine have to go together because they both require 3 hydrogen bonds. Thymine and adenine both go together because they only require 2 hydrogen bonds. And that's the way it'll go. Because uracil can replace thymine in RNA, uracil itself also looks for 2 hydrogen bonds as well. Well, why is it important that these bases always pair up the same way? Why can't adenine go with guanine? Why can't cytosine go with thymine? The simple answer is because having those A with T, C with G allows the structure of DNA to remain the same in every single organism. There is no variation in the terms of what DNA structure will look like. So it gives the structure of DNA order. If we didn't have this order, we'd have irregular structures, which would prevent DNA from packaging up really tightly and would hinder its biological function. That base pairing there also allows us to easily separate the strands of DNA so that we can take DNA replication into account. DNA replication we'll look at as the third video of this PowerPoint.